A couple of days ago, I found out, as you guys probably did from the news, about the new mutant strain of coronavirus, and I wanted to figure out what the excitement was about because um, viral mutations are not unusual. They're kind of the norm, and this virus has been mutating since day one because there's no real pressure for viruses to have genomic stability. They just kind of constantly have mutations, and the mutations that are favorable for it kind of propagate throughout the disease course as it spreads between people. And then the mutations that make them non-functional just disappear. So this is an interesting website called GIS Aid, which was set up back in 2008, mainly to look at mutations in the flu virus. And so everyone in the world, when they sequence a flu virus, they upload it to GIS Aid, and then everyone can look at it, and it helps further research and collaboration and do these things. When COVID-19 started, they put a little part for COVID-19, and so this is their COVID-19 submissions, the SARS-CoV-2 virus sequence submissions. And my internet is slow today. And basically, there's a phylogenetic tree of all the different mutants that have been uploaded to the website. And as you can see here, there's 33,611 different mutants that have been input into here. So mutants are not an unusual thing. And someone took this data from GIS Aid and found out that if you count all the different mutations, there's 25,000 mutations out of the 140,000 different genomes that have been uploaded into there. And then the COG, this is the group that actually discovered the new variant that's in the news right now. It's the COVID-19 Genomics of the United Kingdom Consortium. And basically, if you look here, they say that they have found 1,777 different amino acid changing mutations in the spike protein throughout this. So anyways, this just goes to show that mutations in the virus are not unusual. But something is a little more unusual about this virus, this variant, which they're calling the variant under investigation of 2020 month 12 variant 1. And so this is the minutes of the meeting of the NERV-TAG, or New and Emerging Respiratory Virus Threats Advisory Group. And this was a couple of days ago, December 18th. So they basically look at this new variant under investigation, and from what their conclusion after looking at reviewing the data amongst this group is that they think that the growth rate of this variant is 71% higher than other variants. And they base that based on how quickly the variant is increasing as a dominant strain in the genomes that they're sequencing as surveillance throughout their country. They also looked at R values, and they think that the R values seem to increase as the proportion of this variant increases. So they think that the R value increases somewhere between 0.39 and 0.93 as a result of this variant. Now, R values are a function of the virus and a function of the activity of the infected population. So you can have a very infectious virus that if the population isolates, the R value will fall to 0. Whereas you can have a not very infectious virus where if the people are just really passing it between each other, the R value will go up. So R values are not solely dependent on the virus either. And then PCR-CT values of about 2, and then viral load suggests that the viral load is increased in the new variant. So those are things that are kind of interesting. They think it's more infectious, basically. This thing seems to be more infectious, and that's why they think that that it's something important and something they need to look at. And that's why it is a variant under investigation, because if it's not very important, they feel like they don't even need to investigate it. So this is a something that they felt like was important enough to investigate. There's a couple of things that they decided that they don't have enough data to figure out. So underlying mechanisms of increased transmissibility. So they don't know why is it. It seems to be more transmissible based on their epidemiologic data, but they don't know why. They don't know the age distribution of the cases. They don't know if it's more severe or less severe, and they don't know if this mutation is enough to give it antigenic escape, which means does this change it enough that like the current vaccines would not work against it, or that current people that were infected with the old variant, now has this changed enough that they can get reinfected by it? So they don't know. That's what they don't know. Okay, so I want to mainly go over these two documents, but before we go through those two, I think we need a little bit of background on the structure of the virus to understand those two documents better. And so this is the genomic structure of the virus. And so it has these open reading frames. This is the spike protein, envelope protein, the nucleocapsid protein, and the membrane protein, and other open reading frames here. And then the protein that people are most interested in is this spike protein. And so we'll go to this paper, which talks about, this is when they first kind of characterized the spike protein. This was published in August. And it basically shows that the spike protein is a trimer. Let me see if I can make this a little bigger. It's a trimer. So you have three of the same thing joined together. It becomes a trimer. And then there is the S1 and the S2 region. And this ball right here is the receptor binding domain. And what it does is it binds the receptor, which in people is the ACE2. And then once it binds, it undergoes a change. So like this trimer is kind of like a little machine. It's like a lever. And when it binds this thing, it folds and it pushes the virus onto the cell membrane. And that's how the virus fuses with the cell membrane and gains entry into the cell. And then once it gains entry to the cell, then it releases its RNA, and then the RNA gets transcribed, and then it becomes a virus. So this is very similar to how the new mRNA vaccines work. The nanoparticle fuses, and then the mRNA is released, the mRNA gets transcribed, except that in the vaccine, it only has a portion of the spike protein and none of the other proteins, so it can't make a virus. It just produces the spike proteins cells, and then that gets presented, and you get you develop immunity to the disease. Okay, so that's kind of the background that we need for understanding these two things. Okay, so this is the not a paper, but a communication that they put out, which is preliminary genomic characterization of the emergent SARS-CoV-2 lineage. And so basically, they've been following this cluster that they're calling B.1.1.7, and for the past 4 weeks, and they've realized that this cluster is kind of taking over and is growing rapidly. And so they found that there's a couple of mutations in the spike protein, which they think are going to be important. So let's skip the background. Okay, so here's the kind of the nuts and bolts of this variant. So what they're saying is this lineage carries a larger than usual number of genetic changes. So usually, most variants have 1 or 2 changes. This one has 14 changes. It actually has 17 changes, but 14 of them actually change the protein. So here we go. Yeah. So yeah, there's 4 non-synonymous mutations. So that means there's 14 non-synonymous mutations. That means there's 14 mutations of the RNA in the virus that change the codons. So if you remember from biology, the RNAs have 3 different sequences, like sets of 3 that form codons that basically tell it which amino acid to insert into the protein. So there's 14 of them that cause changes in the amino acid. There's 3 that cause deletions, and there's 6 mutations that even though the RNA has changed, it did not change the amino acid that's specified in the protein sequence. So there's 14 plus 3 that make changes, and there's another 6 that don't make any changes. So a total of how many mutations is that? 17, 23. 23 mutations. Okay, so then this is just an interesting thing that they say, like, why did someone have why did this variant have so many different mutations? And so they think, this is their best theory, is they think that someone who is immunodeficient or immunocompromised was chronically infected. So they basically were infected with SARS-CoV-2 for a long time. And during that time, the virus can replicate many times within one person and accumulate lots of mutations because it undergoes replication without any DNA repair mechanisms, because they don't have any DNA repair mechanisms like our genome has. And so if someone has it for a long time, then they could accumulate a lot of mutations. So that's kind of what they're thinking happened in this case. So this is the part that we're probably most interested in, is potential biological significance of these mutations. So the most important thing is that this this mutation at amino acid number 501. So if we go here, let me see. Does this tell us the sequence? Yeah, so look right here. It basically tells us that this spike protein is 1,273 amino acids long, and it's divided up into like a signal sequence. The first 13 is a signal sequence that tells the cell where to transport the protein after it's been translated. So it's kind of like a like an address, like send it to this area. Then there is the N-terminal domain, and then this is the receptor binding domain. So this is the part that interacts with the ACE2. And then these are the like the transmembrane domain and the cytosolic domain. These are the H1, HR1, HR2. These are these little tails, right? HR1, HR2, right? Okay, so the important mutation is at position 501, which is somewhere like around here. So at that location, there is a amino acid substitution. I think this is alanine to tyrosine. Okay, I don't remember the exact abbreviations, but you can look those up. There's a one-letter abbreviations for amino acids. So I think this is alanine to tyrosine. But when you change that, it increases the ACE2 receptor affinity. So it makes it a better virus. It attaches better to there. And then there's a deletion here. This is a very common deletion that's been seen in multiple other variants. But they don't know if this has any biological significance. So here's a full list of the mutations. So there's a couple of mutations of the open reading frame 1, then the spike protein. So this is the one that they think is really important because this is in the receptor binding domain. They don't know the significance of these other ones. They suspect there isn't much. And then this is another significant one where the open reading frame 8 at the position 27, it became a stop codon. So basically, this open reading frame usually encodes a protein that is usually 121 amino acids long, but now it stops at 27, which probably makes it a non-functional protein. So it makes whatever was the protein that was encoded by open reading frame 8 non-functional. And there is some data to suggest that this makes it a milder infection. It doesn't elicit as strong an inflammatory response if this is a non-functional protein. So this is why you've read maybe that this variant has milder disease, because they suspect that this stop codon makes it a milder disease. Okay, so 2 days ago, the European Center for Disease Prevention and Control put out this threat assessment brief, and this kind of recapitulates a lot of the information that was given in this preliminary communication. So here they kind of talk about the event background. So what we talked about of how this thing is increasing in prevalence along the UK samples. And this graph right here shows as the weeks go by, this is the, they're sequencing, several thousand isolates per week. And as a percentage, you can see that this variant is exponentially increasing. Now, at last time they checked, it was like week 47 was like 11%, but you can see the curve is exponentially taking over and becoming a larger and larger proportion of the variants they sequence. So they feel like because this thing is increasing at a time when prevalence of the virus overall in the population is pretty high, there must be some advantage. This virus is somehow a um, better at infecting people than other variants, and that's why it's taking over. Because if it wasn't better, it would be just very flat. And then genomic properties. This is what we talked about. Here's a phylogenetic tree of the variant, much like the phylogenetic trees we saw in GIS-AID. Okay. So I think the important things are here. Okay, potential impact. So they're saying basically this deletion makes it so that some PCR tests will not detect it. So they're saying you should not use PCR tests that primarily depend on the spike protein gene sequence for the PCR test. These should have multiple because this will miss it. So they're saying that they don't, again, this is information they don't know, and they don't, there's some reason to believe because of that deletion in the open reading frame 8 that it might be milder disease, and that's what they're talking about here. And so far, none of the variants that we know about cause more severe disease. And so this is what they don't know also, like mutations in this new variant may alter the antigenic properties of the virus. And so that means that two things. One, it may mean that the vaccines that are rolling out now, which are based on the old sequence, may not work for this virus, this new variant. And it may also mean that people that have been infected with the old strain might be susceptible to reinfection with this variant. Now, there's a couple of reasons to believe that that may not be the case, and I think we'll know the answer to that pretty soon. Moderna has already said that they are going to grow this new variant and test neutralizing antibodies uh, from their vaccinated patients and see if it identifies and neutralizes this virus. And so we should know the answer to that in a few weeks. They said that in 2 weeks they would know the answer to that. And the reason that it's probably going to be okay is because the spike protein is big enough that it has multiple epitopes. So there's multiple areas where an antibody or a T cell receptor could recognize it. And so just changing, so there may be a few antibodies that you create that don't recognize the new variant, but there's enough conserved sequences that it would probably recognize it still because it hasn't changed that much. But we'll know the answer to that in a little bit. And then they'll also, if there's a lot of reinfections, we'll know that that this is a problem. If there's no increase in reinfections, then we know that's probably not a problem because that's what they talk about here. Okay, so in summary, there's been viral mutations are extremely common. They've been happening all the time. This one is special because it has a lot of it has more mutations than is typical for a variant that accumulated all at once. And then there is a change in the spike protein at position 501 that seems to make it a better match and have tighter affinity to the ACE2, which makes it possibly a superior virus for infecting people. And so it does seem that it is more infectious based on epidemiological data. And then lastly, we still don't know whether it's going to change the antigenic properties of the virus enough that it will make the vaccines not useful. But there's reason to believe that that won't be the case, and we will know a kind of a definitive answer in about 2 weeks. So thanks for watching.